All right, good morning again, Dr. Cotton. You're reminded, ma'am, you are still under oath, and Mr. Clarke, you may continue with your direct examination.
Thank you, Your Honor. Good morning, ladies and gentlemen. Dr. Cotton, as far as these methods of DNA typing, and you've talked in broad terms about PCR and in more detail about RFLP, are these techniques simply a better way to type stains than, for instance, ABO typing or conventional serology?
Yes, they generally will offer more information than conventional serology will offer.
Again, we spoke about RFLP, or you spoke about RFLP typing yesterday. The second technology that you mentioned briefly yesterday, I believe you referred to as PCR, is that right?
That's right.
What year was that process discovered?
I believe that the first paper on PCR was published in 1985.
As far as its use in forensics, is it newer or older than the RFLP typing process?
It's actually it came along at about the same time, although it was not being used quite as widely early on as the RFLP process.
Are you familiar with who was the first scientist to use this PCR process in forensics?
Yes, I am.
Who was that?
That would be Dr. Ed Blake.
Do you know approximately when that forensic use of that technique occurred?
I only know that it was early, maybe 1986 or so.
As far as this PCR process, and many of us, not all of us, have heard of the movie Jurassic Park, what does PCR have to do with that?
Objection, Your Honor.
Sustained.
PCR does what? Can you give us a very brief summary?
PCR is a way of saying, I want to replicate a small section of DNA, and it allows you to replicate a small section of DNA in a way that if you have a very small amount of starting material, you can go through sufficient number of replications so that by the end of the process, you will have enough of this small section to then do some analysis on.
What role does it play, for instance, in old samples? And let's first of all ask, can this process be used on samples of DNA that are perhaps years or decades or centuries old?
It can, and it is being used on those types of samples.
What types of samples is that?
There are samples from, I don't know what the proper term would be, but very ancient, for example, bone samples that have had very short sections, copied using PCR to look at how genes have changed over time. That is, if you can have a sample from a human being who is several thousand years old and look at a particular sequence of the DNA, you might say, well, how does that sequence compare to the same sequence in modern human beings?
Has PCR been used on ancient animals?
I don't really know. It may have been, but I don't really know.
As far as this PCR process, is it like RFLP in the sense that it's a testing method, or is it something a little bit different?
PCR is simply the process that allows you to make a lot of copies of a particular section of DNA. And then the rest of the analysis may have many different forms. That is, what you do now with your section of DNA that you've replicated many times may not be the same for every purpose or every question.
Would it be correct then that PCR would be more like one step in a typing process when DNA is being typed?
Yes.
What about your own knowledge of PCR? How did that come about?
I had been to several meetings where PCR was discussed. I had done some reading where PCR was discussed. My own hands-on experience came from visiting 2 different laboratories where they were actively doing PCR and learning from the staff in those laboratories how to do PCR. And both of these visits to other laboratories. One was 2 weeks and one was a week. I had an opportunity then to learn from the staff there, to do it myself, and then going back to Cellmark to help the rest of the Cellmark staff, who also had, some of whom had also been out to get training, which was different than mine, to set up the PCR that we do there.
Who were the individuals that you, whose labs you visited to learn PCR?
I went to a laboratory at Houston. That was Dr. Tom Caskey. And I went to Dr. Alec Jeffries' lab in the UK.
The same Dr. Jeffries that you described yesterday?
Yes.
How about your own personal hands-on experience with PCR? Could you describe that, please?
Well, I had those 3 weeks, and then I had some experience, hands-on time, doing the PCR reaction while we were in the process of doing validation studies to set up PCR in our laboratory, and a number of other people did too. I am not doing the PCR test now. The PCR tests are done by the regular lab staff.
As far as this copying process, is there a term used to describe what you actually do in taking small segments of DNA and copying them over and over?
Yes, the term is amplification. So people will talk about I've amplified a certain section of DNA.
This process, and I believe you said that this process of copying over and over is directed at short segments of DNA?
The segments that are used for forensic analysis are relatively short, and I'm talking about perhaps 100, 200, up to 400 base pairs in length. That's a pretty, in DNA terms, that's a pretty small piece.
So you're not talking about copying over and over this entire segment of DNA that you described as containing, what was it, 6 billion bases or so?
No, you're not copying the whole thing. You're just copying a particular section.
With regard to this copying process, would it help to use a chart or a pad so that you could draw what this amplification or copying process involves?
Sure.
All right. Your Honor, then with the court's permission, Mr. Fairtlow will, I believe, set up the drawing pad for the witness.
We'll reassemble our drawing pad.
Reassemble.
Yes. And Mr. Clarke, I noticed yesterday that you were using the dry erase type marker. Does that have a longevity to it, permanence to it?
I hope it's not disappearing ink.
You notice that's what you're using there is dry erase stuff.
One over there. The dark blue one, I think, is just a different.
This diagram be marked as people's next in order, which I believe is 247.
247.
All right, Dr. Cotton, if you could step down from the witness stand, and what would be an appropriate label for this chart to aid you in describing this copying process or amplification?
We'll just call it amplification.
Very good. If you could just write that at the top. All right, now if you would go ahead and if you could describe for the jury, please, what is this copying process?
Okay, keep in mind now that you're starting at have a sample and you've extracted the DNA. So those steps are the same as, or similar to, the RFLP process. And so let's just say, for simplicity purposes, that we have a single chromosome's worth of DNA that I'm going to use as a reference point here. So we're going to put in the tube our starting DNA, and we're going to put in the tube a supply of the 4 DNA components: A's, T's, G's, and C's. And we will also put in the tube what an enzyme, this is a protein, and it's called a polymerase. And this polymerase is basically going to assemble the A's, T's, G's, and C's. And we're going to put one more thing in, which is referred to as a primer. And that's a very small piece of single-stranded DNA, usually about 20 bases or so. It can be longer or shorter, but that's just an example. So that we now have the components to make more DNA. This is what we're going to use to make more. We need our starting material, we need the building blocks, we need something to put the building blocks together, which is the polymerase, and the primer serves as an anchor for that polymerase to hold on to.
Let me stop you for just a moment, Dr. Cotton. With regard to this term polymerase, is that the first word of PCR, the process itself?
Yes.
And what does it stand for again?
Polymerase is a protein which can assemble more DNA in this case.
And what does PCR actually stand for? What are the words?
The PCR stands for the polymerase chain reaction. And basically, you're going through a cycle of steps, which is why it's referred to as a chain reaction. You're repeating a cycle of steps over and over and over again. And what we'll do here is just show you what 1 or 2 cycles looks like.
All right, go ahead.
So the first thing we'll do is heat this DNA back up to about 95 degrees, enough so the strands will separate.
Again, is that centigrade, not Fahrenheit?
Yes. And at that point, we'll allow the short primers or anchor points to bind, and these primers are designed so that the sequence of DNA that they have to hook up to is known, and so you have a single-stranded piece in the primer binding to this now single-stranded starting material, and all the A's and T's and G's and C's are pairing up. And then the polymerase will come along and, directed by the bases on your starting material, will reassemble a second strand.
All right, Mr. Clarke, we have a vision problem here.
Yes. Or, Your Honor, would it be possible to put the drawing pad here where the podium is?
Let's put it there. Why don't you turn it as directly perpendicular as you can. Thank you.
I'm not sure we've solved all the vision, though.
All right, you're number 1. Can you see that? All right, thank you, counsel. Proceed.
And if you could, Could you briefly describe again what you've already described, and perhaps you can point to the various portions of the chart so that each of the jurors may see it?
We have double-stranded DNA starting material, which we're going to heat. And the two strands, we'll just put a little X here on our starting material strands. Drop the temperature down, allowing the primer to bind, the two primers to bind, and then allowing the polymerase to make a copy of this starting strand. So the polymerase is basically going along and says, I have a T on my starting strand, I'll put an A in. I have a C on my starting strand, I'll put a G in. I'll have a G on my starting strand, I have to put a C in, and so on. So now where we originally had a single strand to start out with, we now have two. Now
Go ahead.
I'm sort of going to run out of a length of paper, but the idea is that you then start the process all over again. So you go, just stand on this side. So you go, start the cycle again, heat it up. These two double strands will come apart. These two double strands will come apart. And now again you drop the temperature down and let the primers bind. Now I've just made these shorter because there's not enough room. They wouldn't really be shorter, they'd be the same length. And then allow the polymerase to finish off making the copy for each one of those. And so now we have 4 completed strands where we only started with 1. So as you can see, each time you go through the cycle, you're going to double the number of strands that you started out with. If we start out with 1, then we would go to 2, and then we would go to 4. The next cycle, you would go to 8, and so on.
All right, and perhaps you can stand on the other side so that I can ask you a few questions about this process. You've used the term primer. Does it act as something like a primer in priming a pump, for instance?
Well, it's not a bad analogy. It's not great. What it is the characteristics of the enzyme are such that it needs an end to start from. If you just separated the strands, it wouldn't have a place, it doesn't have an anchor point. So the primer is serving as an anchor point for the enzyme to then connect the next base to. It needs something in the way of the start of a second strand to actually connect. So the primers are in there both to say, this is the section we want to copy. That's really what the primer does, is it identifies the section that you want to copy, and then it serves as an anchor point for that enzyme to sit down and then move through and copy that piece of DNA.
How do you know where the primer actually attaches to this segment of DNA?
You have designed your system, that is, you have to know up front the sequence of bases of the piece of DNA that you want to copy. There has to have been enough research done and development done so that, but at least in the forensic setting here, you must know ahead of time the sequence of bases that exist in the piece that you want to copy. That has to be done ahead of time. Then you can manufacture in the lab the primers that have the sequence at the beginning and the end, and then you're set to go.
As far as these known sequences of where it starts, for instance, and you mentioned you have to know the sequence, have scientists looked at these areas to determine their sequences?
Yes, the development work for the genetic locations that are used for forensic testing with the PCR methodology, these regions have all been sequenced, and a lot of other work besides the sequencing has been done to study these regions before applying that information to actually designing a forensic test.
This copying process, first of all, do our bodies do that by themselves?
Not with this particular polymerase, because this polymerase is able to work at a very high temperature. But all our bodies have a similar enzyme so that when a cell replicates and divides to form two new cells, in the process of doing that, you are replicating the DNA, and the enzymes that perform that replication are also called polymerases.
Would it be correct to say then that this PCR process simply does outside the body what the body does on its own?
Basically, that's right.
And it's as simple as that? In terms of this division and copying and so
Yes.
forth? All right. Now, Your Honor, if we could, I'd like to put that drawing pad back where we had it yesterday and then continue on with some additional prepared charts. All right. And perhaps you can have a seat again on the witness stand, Dr. Cotton, but probably not for too long.
That's not so good. I can't see you.
Now, as far as this use of PCR, and I'll step over here, Dr. Cotton. As far as this use of PCR in forensics, to your knowledge, do we have some charts, prepared charts, that illustrate how you actually use this technique to type samples in forensic casework?
Yes, I've seen the charts that you're referring to.
All right, Your Honor, at this time I'd ask to be marked as People's Next, which I believe is 248. Record, this diagram can be labeled or can be described as PCR analysis, and then showing at the top two large blocks, step 1 extraction and step 2 amplification, and then a smaller block labeled step 3 detection.
Yes.
With regard to this chart that's been placed on the board, have you had an opportunity to look at this before this morning?
Yes, I have.
And with regard to this chart, does it illustrate various steps in the PCR-based typing process in forensics?
Yes, it does.
Now, in particular, well, are there a number of steps that occur in this typing process?
For sure.
In general terms, are there fewer steps than in the RFLP typing process that you described yesterday?
Oh, there might be fewer. It sort of depends on how you count them.
Okay. This board itself, and it's labeled at the top, step 1 extraction. Can you describe what that refers to? And if there's a pointer there that would help you, please feel free to use it.
The top rectangle is simply illustrating that you have some kind of a stain, and from that you do a DNA extraction. And the DNA is going to end up basically in your test tube. And these tubes are actually very small. And then to that tube, you will add, in addition to adding your DNA, you'll add what's diagrammed up here as the PCR mix. And the PCR mix is the DNA components, the A's, T's, G's, and Z's, the polymerase, and the primers, and some other salts or buffers. So all of those things go together in a single tube.
All right. What happens at that point?
At that point, you take that tube and put it in what's called a thermal cycler. It sort of looks like a cash register, actually, or a small one, but it has a metal block, which is illustrated right here, and a computerized, programmable method for controlling the temperature in that metal block. So the tube sits down very tightly. The holes in the block are basically the same shape as the tube, so that when you put the tube down there, there's close contact between the tube and the walls of the metal block. And the temperature in the tube is then controlled by heating or cooling the metal block, and you do this in a cyclical manner. And the cycle goes, as we talked about a little while ago, you're heating the block up to 95, so the contents of the tube will be at 95, so you can denature the DNA, separate the strands. The temperature is then dropped down, and I'm not going to be able to remember what the exact temperatures are in the cycle, but the temperature is dropped down somewhere usually around 60 or 64, 65 degrees. Could even be lower than that. Anyway, I don't remember right now. To allow the primers to bind, and then the temperature is raised up to around 70 degrees to allow the enzyme to complete copying of the strand, and then the cycle starts all over again. So you tell the machine in your programming what specific temperatures you want it to cycle through and how many cycles you want it to go through.
In other words, this machine, you can program it or tell it basically how many of these cycles to go through, at what temperatures, and so forth.
That's right. And how long each cycle is to be. Like, you're going to leave it at 95 degrees for 1 minute, and then you'll drop it down to 55 degrees and stay there for 30 seconds, and then you'll raise it up to 70 degrees and you'll stay there for 50 seconds or something like that.
This heating and cooling process that you described, was PCR, for instance, used before there were machines that did this in a fairly automated fashion?
It was, and my understanding was when PCR was developed that basically the people were doing this by hand. That is, holding the tube in a water bath at 100 degrees and then taking it out and putting it in another water bath at another temperature. So it would have been very tedious to do that. You'd have a lot of researchers standing around taking up a lot of their time holding their tubes in water baths. So anyway, the computer age came, and this machine was developed to essentially take care of that for you. So you put your tubes in, you set the machine up, and turn it on, and it goes through as many cycles as you've programmed it to do, and then it holds your tube at a cool temperature until you come and retrieve it.
Just briefly, there's a step 3 listed that's called detection. Is it your understanding that we'll return to that in some detail in just a few moments?
Yes.
All right, with regard to this amplification process, and in particular how these strands are copied, would it aid you to use a prepared diagram showing 2 different double helixes or ladders?
that only really relates to the kind of detection that we're
All right, very
doing,
good.
so we could leave that for,
As far as the detection itself?
yes.
Okay. Then if I could, I'd like to return to the drawing pad. And Dr. Cotton, could you use the diagram? And let's add a new page if we could that I believe would be People's
249.
249. And with regard to this, if you could just illustrate, and I'd like you to actually write down a series of, just for illustrative purposes or example, bases, and how in this copying process, how the primer would then act to add these bases that are floating around in this tube mixture. Do you understand what I'm asking? Okay.
You want me to take a double strand, take it apart, and show how the second strand is
Correct.
reconstructed?
Just one cycle's fine. And perhaps you could lay, what would be an appropriate title to illustrate that point?
More amplification.
Okay.
Oh, sorry, I didn't hear the answer.
More amplification. It's going to take me a minute to set this up.
Very good.
Okay, so I've made a very short sequence, and I see there's a flaw in my thinking here, but we'll sort of try to work past that. Double-stranded, A's and T's, G's and C's nicely paired up. Heat it, and create the two single strands. We're just unzipping the zipper. And now we want our primer to bind. So here's the flaw in my thinking. I'm going to have to make for a very short primer because I didn't draw a very long sequence. So let's say we have put in a primer here to have two T's, and we put in a primer to fit the other side, which is going to be a T and a G. And you can see that this primer won't attach to this side, and this primer won't attach to this side, and the primers won't attach to each other. So you have to take these things into consideration. And then you simply have your enzyme come along, and the enzyme will come along and create this new strand. It will use this primer as its anchor point, and now add, and remember we've got, just individual A's, T's, G's, and C's floating around here available to be put into the new DNA strands. So the primer will come along and correctly make a brand new strand on both sides.
And what you...
And I said primer, but I meant polymerase. The polymerase will come along, hook onto this primer, and correctly finish off here the new strand.
So what you've illustrated is basically how this process works with the use of the primers, and how from one strand you create two strands.
Yes.
And then that process just continues through the cycles, the number of times over and over that you described a few moments ago.
That's right.
All right. Then, Dr. Cotton, if you would, and what I'm going to ask you is, with excuse me, Army, I have a moment. couple more questions, if I could, Dr. Cotton. With regard to this primer, it latches on to the other side of the DNA strand, is that right?
It will bind, there is no other side. This is a single strand now, and this is a single strand at the point that you've heated it. So the primer is the first starting point to creating another side.
And it's from the primer on, up, or down the ladder, as the case may be, that it then starts this process of adding these bases one at a time.
That's right.
All right, very good. With regard to, and now just referring very briefly to the large chart, which I believe is People's Exhibit 248, as far as this, and you have described the second step on this chart, amplification, is that right?
That's right.
With regard to the third step, is that the final basic step in this process of PCR typing?
That's right. You're going to, now you've made lots of copies of your DNA, and you want to look at it. For purposes of forensic use, you will be copying sections of DNA that have some difference from person to person. And now you want to figure out, well, what does that difference look like for the sample that I just copied?
In other words, you actually obtain the types in a particular sample so that you can determine, for instance, what persons can be excluded or included as possible donors.
That's right.
With regard to this detection process, is it the same or different from what you're looking for using the RFLP method?
Depending on the genetic location that you've amplified, there is one of the detection methods is similar to the RFLP. That is, you're simply looking at how long a piece of DNA you amplified. You know you started at one end, but there's some variation in the length, and we may need to explain that a little bit more. But the other type of test that's available does not have anything to do with length, really, but has to do with a sequence difference. So that at a particular genetic location, I might have one sequence, Mr. Clarke might have another, Mr. Goldman might have another. And so you could look at the sequence differences.
Do we have a board that will illustrate that difference as well?
Yes, we do.
All right. Your Honor, I'd ask that a prepared diagram be marked as
Equals 250.
Thank you. Your Honor, for the record, People's Exhibit 250 could be described as labeled DNA with what appear to be two depictions of a ladder side by side, DNA ladder. All right. Dr. Cotton, with regard to this exhibit, first of all, have you had an opportunity to see it before?
Yes.
Would this exhibit help you describe the differences between these two methods of typing samples that you just made a brief reference to a moment ago?
Yes, it's the good example for, and it's a point of discussion for what is a sequence difference.
All right. Could you then use this diagram to discuss that?
Yes. What we have here is the same diagram basically that we were using yesterday to show what is a DNA molecule. And here we just have two of them that are side by side. And if you just go, let's say, from the top to the bottom, on one you'll see we have an AT pair at the top, and the other there's an AT pair at the top. Then we come down to a GC pair, and the second one also has a GC pair. But here in the middle, the DNA molecule on the left has an AT pair. DNA molecule on the right has a GC pair. So, and then if you go on down the remainder of the three base pairs here, they're alike, a GC, an AT, and another GC. So the only difference we have here is the fact that the third base pair down on one diagram is an AT and on the other is a GC. This is a DNA sequence difference. This sequence difference can be detected because you can amplify both of these pieces of DNA and then use a detection method to allow you to match either these pieces or those pieces, and you can figure out that you have two varieties basically in your sample. You have one molecule that has an AT pair and the other one that has a GC pair. And so in the PCR diagram that we were looking at a little while ago, when it got to the bottom and it says detection, this, to amplify two pieces of DNA and then detect that they had a sequence difference, is one type of detection that's used for forensic analysis.
What's the other type of detection method that the board just named very briefly?
The other type is looking at a length difference, which is very similar to what you're doing in RFLP. You're also looking at a length difference. But maybe we should go back to the piece of paper just very briefly.
You're going to create a new diagram, Dr. Cotton?
Yes, I am.
All right. And Your Honor, for the record, the witness is labeling this as PCR length difference.
All right, this will be 251.
What I'm going to do is go back very briefly to the same analogy or the same example that I used yesterday in terms of a repeat. So let's say we have the same CAT repeat, and I'm just using that because it's a very easy example. And I have two different DNA pieces here. And one has 2 repeats and the other has 4. And again, if I can somehow identify the ends of each of these pieces, I could look and see how long they were. For the RFLP test, we defined the ends by having an enzyme come along and literally cut these sections out. You can use the PCR test to also look at the lengths, and you can do that by designing your primers so they will come along and bind just outside the repeat. Now, we could go through and redraw all the amplifications, but if you think about, if we look at just one of the strands here, our primers come along and copy this section and copy this section. Ultimately, as you go through the PCR, you will end up with a piece, many pieces, that are this long. If you do the same thing with the other piece, with your primers sitting just outside the repeats, the pieces that are eventually going to become your product, or your many pieces of DNA that you've now copied, will include the full length of the primers and the 4 repeats that I've diagrammed, whereas over here we only had 2. Now, instead of having a sequence difference like the board that we just talked about, now you have PCR product, but the difference is a length difference. And you can analyze that length difference by separating the DNA strands on a gel. It doesn't happen to be exactly the same kind of gel we talked about yesterday, but the principle is exactly the same. And that is the DNA strands will move through the gel and separate out according to how long they are. And that is the other commonly used method of detection for looking at your PCR product, that is looking at some kind of genetic difference in the DNA that you've now amplified.
As far as the sequence differences, is my sequence difference at a particular set of markers different than yours?
It may be, and it may be the same. Since we haven't tested both of us, I couldn't answer that question.
What about in the area of these length differences?
The same thing. The variation in the population for these PCR markers is not extensive. So many of us may share some things, and then some of us would be different. So whether or not you and I are different, I couldn't tell you.
And that's
we
why...
may be, but I don't know.
Do you look at multiple genetic markers, more than one genetic marker, to determine these differences if they exist?
You look at as many genetic markers as you have the ability to look at, and no matter what kind of system you're using, be it PCR or RFLP, the more markers you look at, the more genetic locations you analyze, the more information you have. That's a generalization that will always be true for forensic testing. The more markers you look at, the more information you have.
And is that your goal as a forensic scientist, to look for as much information as possible?
Generally, that would be the goal, sure.
All right. I have, first of all, to your knowledge, do we have one more board to illustrate this PCR typing process?
Yes, I think so.
Okay. Your Honor, at this time I'd ask that be marked as people's next in order
251.
a final PCR chart.
I'm sorry, 252, 251 being the drawing.
Your Honor, this exhibit.
252.
252. It's also labeled PCR analysis, but it has 2 small blocks at the top, step 1, step 2, and then a large block, step 3, for the record.
All right.
Now, Dr. Cotton, with regard to this chart, People's Exhibit 252, would this help you in describing how these actual differences, whether they're sequence differences or length differences, are determined by you as a forensic scientist?
Yes.
All right, then if you would, with this chart, then describe for the jury these differences in typing and how they work.
Okay, so 1, we extracted the DNA. Step 2, we amplified it. We used the thermal cycler like you saw in the previous diagram. And now, based on whatever genetic location we amplified, we're going to use one of these two types of detection. And the first type that's illustrated, which is on the left, is called a dot blot or sequence polymorphism. That is, sequence polymorphism just meaning sequence difference. It's exactly what we looked at on the 2 DNA molecules just a few minutes ago. So the setup is you have a nylon strip which has DNA bound to it in specific locations, and that's illustrated right here. There's a plastic tray, and the tray has like little wells in it, sort of, and the strips sit in the tray. Solution is added to those strips, and your amplified DNA is added to those strips. And the DNA that is spotted onto the strip will bind to whatever part of your amplified DNA that it matches up with. And where the DNA binds to the strip is then processed through a series of reactions to give you a blue dot on the strip where your sample DNA bound. And these blue dots are then interpreted to say you have, this is a term we haven't used. When I talk about genetic differences, if you go back to just mother and father, if you have 2 differences, those are referred to as an allele. An allele is simply a form of a gene. So that, for example, the 2 sequence DNA differences that we showed just a few minutes ago, those could be called 2 alleles. Or the term Mr. Clarke was using would be 2 types. So from the series of blue dots on the strip, you will read off the types or the alleles for that DNA sample that you just analyzed. And then you simply write those down and record those. The other method, which is diagrammed on the right.
I'm sorry, let me stop you for a moment, Dr. Cotton. And Your Honor, I'd ask that be marked as people's next in order what can be described as a tray containing various slots in it.
All right, 253, plastic tray.
Dr. Cotton, showing you what will be marked. I'm sorry, was that 253 you're on?
253.
What is that item you have in your hands?
The item is the typing tray. It's the tray that's illustrated in the top 2 pictures here. It's plastic. This tray has strips laid in the wells. The strips have not been processed, so there's no blue dots on them. And the tray has a clear plastic lid, which I taped down so that I wouldn't lose it. And this is the typing tray that's used in the laboratory, and examples of the strips that are used in the laboratory to go through this series of reactions of adding your amplified DNA and developing the blue dots on the strip. And from those blue dots, then you would read the types from those samples.
Where did that tray actually come from as well as, and I'm sorry, did you say there were strips inside the tray right now?
Yes, there are.
Where did they both come from?
They came from my lab, but they're manufactured by Clarke and Elmer Cetus, I believe.
Would that be a tray and strips then that you would have used in casework if it hadn't been brought to court today?
Oh, sure.
With regard to those strips, and they're currently laying in the bottom of the tray, is that right?
That's right.
What else is put into that tray other than the strips?
You want to go through the series of things that goes in here?
If you could, yes.
The strips go in first, and a solution that has some salt in it goes in, and your amplified DNA goes in, and that's incubated in a water bath. So the water bath is controlling the temperature. The water isn't coming over the tray. long enough for your amplified DNA to bind to the DNA dots that are on the nylon. Then a series of reagents or components are added which allow, well, first the extra DNA is washed off, and then a series of reagents are added to allow the development on the strip of the blue dot wherever your DNA bound. So you go through adding your amplified DNA, pouring off any that didn't bind, adding in a series the reagents that you need to develop the blue dots, pouring that off, and then allowing them to develop.
And then finally, do you see the appearance of these blue dots as is illustrated near the bottom left-hand side of People's Exhibit 252, the large diagram?
Yes.
All right, Your Honor, at this point, I'd ask that this exhibit be allowed to be distributed to the jury.
The plastic tray.
Yes.
All right, hand it to Dr. Cotton. Would you hand that to juror number 1, please? All right, Mr. Clarke, would you collect 253 from Deputy Russell, please? And may I see that? Thank you. Doctor? Thank you. Mr. Clarke?
Dr. Cotton, if you could you then turn to the right-hand side of People's Exhibit 252, and does that describe a different detection method or typing method when you're looking at these length differences?
Yes, it does.
All right. First of all, at the top, it seems to have some letters and numbers. What are those?
It reads AmFlip D1S80 length polymorphism.
Okay, what's that first term mean, AmFlip?
AMPFLP is sort of a shorthand version of saying amplified length polymorphism. And AMP referring to amplified F is fragment, L is length, P is polymorphism. D1S80 is the nomenclature for the genetic location that is currently used in forensic labs that has a length polymorphism.
We'll return to that later, but is D1S80 simply a way of describing a particular genetic marker?
It's just the name of the marker.
Like PGM is the name of a particular
Exactly.
protein marker?
Exactly.
All right, go ahead.
Okay, so in this case, you have amplified DNA. The pieces that you amplified may have different lengths, and so you're using a gel, which is our green gel here with the red DNA on it, and the DNA is loaded into the gel. It's a slightly different type of gel than the one I described yesterday, but the principle of how the gel works is exactly the same. And the gel is subjected to electrophoresis, and the current moves the DNA through the gel. It moves through based on its size. And in this case, you don't have to use P32, you don't have to do anything fancy after you've run the gel. You can simply use a stain and visualize where the DNA fragments are. So at the end, you have a gel that's stained, and you can actually see the DNA fragments directly, and then you can photograph that gel, and you can compare the positions of the DNA bands in the various samples that you've now analyzed.
And is that comparison of the bands like that same process of comparing bands using the RFLP method?
It is.
Now, with regard to this length difference and this typing method described on the right, is that again simply one of the means of typing a particular genetic marker following the use of this PCR copying process,
That's right.
as well as this dot blot or method used to look at differences between people where it's based on a sequence difference?
That's right. With the RFLP, everything you're looking at is length. When you use PCR, you can use a test that looks at sequence differences. That's illustrated on the left, or you can use a test that looks at length differences, and that's illustrated on the right.
One more question, if I could, and particularly while you're up, Dr. Cotton. I'd like to take you back to the current drawing, which I believe is People's Exhibit 251, labeled PCR length difference. With regard to that particular drawing, you described the use of these enzymes to cut the DNA at certain locations.
I simply made reference to that in that for for RFLP, to define the ends, that is to define the length, you use an enzyme to cut that length out. For PCR, to define the length, define the ends, you're using your primers. That defines the end, and then you're copying it, however many repeats happen to be in between. And whether you have a few repeats or a lot of repeats, that determines the length.
And these primers, how do they know exactly where to start this process?
The primers don't really know anything, but they are simply a piece of DNA that's single-stranded, and they are going in and binding with the piece of DNA in the reaction tube that matches them. That's why my short primer example wasn't very good, because to be very specific, the primers need to be longer, and they are usually around 20 or possibly more bases.
But for purposes of showing how this process works, that's why you used shorter lengths or shorter sequences.
Well, if we drew out all those number of bases, we'd be here all day.
We need a taller chart.
We would.
All right, very good. Your Honor, it was my intent to clear the charts and enter a slightly different area.
All right, proceed.
All right. Dr. Cotton, as far as the use of PCR in your laboratory, was there a time when you actually began using this technique in your actual casework?
Yes.
When did that happen?
We started doing PCR and casework in about, let me think about this a minute. I believe it was about June of 1992.
And you began using the RFLP process in casework when?
For forensic casework, we began about early 1988.
How do you decide, if a sample or a case comes into your laboratory, how do you decide whether to use this RFLP technique or to use the PCR copying process followed by typing at genetic markers that PCR looks at?
For our particular laboratory, that may be related to a lot of different things. For example, it may be that another lab had already done RFLP but doesn't have the capability of doing PCR, so they might specifically be asking us to do PCR. More typically, we would get a piece of evidence in, and we would have to make an assessment. Is this piece of evidence, does it have enough DNA, and is the DNA in sufficiently good condition to do RFLP? And if so, then you would proceed with RFLP. If the DNA or the piece of evidence contains only a very small amount of DNA, or the DNA is very degraded, then you would choose to go forward with PCR.
That's what I was just going to ask next. Do they each have their own advantages, these two different approaches?
Yes.
Can you describe that? Let's start with RFLP.
For RFLP, you need a larger amount of DNA, and it needs to be in very good condition.
Why is that?
Because the test, if you remember yesterday I was talking about lengths of DNA that were thousands of base pairs long. You're looking at, in our lab, we're looking at a piece of DNA that's anywhere from about 1,500 up to 12,000 base pairs. So your DNA has to be in good condition it has to be much bigger than that as starting material, or your test won't work. So it has to be in good shape. And you have to have a fair amount of it. The RFLP test has the big advantage in that it's very discriminating from one person to the next. There's so much variation in the population that each time you look at another genetic locus or genetic location with RFLP, you're getting a lot of information. That is, you can discriminate one person from the next with a high level of discrimination. On the other hand, the PCR, the tests that are currently available, doesn't have that level of discrimination. It is a much lower level of discrimination, sort of in the range or maybe somewhat better than a whole series of serological markers. However, the PCR can use DNA when you have a very small amount, or it's in very poor condition, or both.
So in other words, in terms of the ability to tell many of us apart as human beings, in other words, a powerful test as far as this ability to, again, tell large populations apart, is the RFLP procedure then the more appropriate to be able to determine that type of information?
Absolutely. Then that would be the test of choice.
As far as the PCR process itself, and let's talk about both techniques in terms of how long they take to obtain results. Are there any differences between the two?
There can be significant differences.
Can you describe that, please?
If you brought a sample into the lab and you didn't have, say you didn't have anything else to do, every other case that you were working on was all finished, and you simply went through the PCR process, you could have results easily by the end of the week. So it'd be very fast, and that's assuming you have, a moderate number of samples, say 3 or 4 or something. Easily you would be done in a few days. The same test with RFLP could easily take you 3 months.
Why is that? Why does it take longer?
The length of the time it takes to do the RFLP test is pretty standard from the time you extract the DNA till you load it on the gel. But the exposure of the X-ray film to that nylon membrane, if you have a lot of DNA, it can be short as a day. But if you have little DNA, it can be as long as 2 weeks. So it could be that for the RFLP test, you took 2 weeks to look at one genetic location, then you took another 2 weeks to look at the second, and another 2 weeks to look at the third, and you can see that rapidly eats up a lot of time.
So it's that exposure or creating this X-ray film that takes a great deal of time, is that right?
It can, yes.
Are there differences in the time it may take to create this X-ray from a particular sample?
Yes, and the differences are basically related to how much DNA you have in your sample. If you think about a large amount of DNA and a small amount on your nylon membrane, you add your P32 DNA probe. If you have a lot of DNA there, you can bind a lot of probe. If you only have a small amount of DNA there, you only bind a small amount of probe. It's the emissions, the radioactive emissions from that probe that expose the X-ray film. So if you have a lot of probe there, you'll get an exposure on your X-ray film quickly. If you only have a little bit of probe bound because you only had a little bit of DNA to start with, it takes a long time to get enough exposure on that X-ray film to actually see something.
And again, if I can show you what was marked yesterday as People's Exhibit 246. Is that one of those X-ray films or autorads that can require this fairly lengthy period of time to develop?
Yes.
Now, you've described a little bit about the use of genetic markers, and you actually identified one of the particular markers. I believe it was D1S80, is that right?
That's right.
Is that just simply science's way of designating markers to tell one from another?
Basically, yes.
Is there any particular reason there's numbers and letters in it instead of just letters like PGM?
The DNA locations that do not code for, do not have information for a gene that we know of, are given letter designations. The D stands for DNA. The 1 is because that particular location is on chromosome 1. And the S80, I actually can't remember what that refers to, but that also has a specific reference. So, and they're called anonymous DNA pieces, that is. They're not a gene that we know about, and they're just given these D designations. It's an international nomenclature, so that if I read a journal article and it has one of these probes, it might say D5S20, and that would tell me that that DNA location was on chromosome 5, and if I could remember what the S20 meant, it would tell me more than that.
As far as these genes that you say have these descriptions, like starting with a D and then a number of the chromosome and so on, you said there something about them was not known. What is that?
Most of the, in fact, as far as I know, all of these genetic locations that contain these repeats, these are not genes in the traditional sense. Biologically speaking, in the traditional sense, a gene is a piece of DNA that contains information that creates a protein that then can go out into the cell and has a function. We know that these repeated sequences are not genes in that sense. Scientists don't actually know what the function of these repeats are. That doesn't mean they don't have one, it just means we don't definitely know what it is.
As far as forensic science, and in this process, do you have a process of deciding or selecting which genetic markers to look at for purposes of identifying people?
Yes, for all of the markers that are currently used, there's been a substantial development period. That is, of the markers that are available, people have gone in and done the kind of research to say how much variation is there in the population for this marker, how easy is this marker to use, how much development do we have to go through so that many laboratories could have access to this particular marker. So this kind of work is done in a development sort of stage, and then a marker is selected because of its utility, its ease of use, its discrimination power, and so on, to be then used in a wider setting with where many labs have access to or may choose to use a particular marker.
As a forensic scientist, would you, for instance, normally be interested in looking at the cystic fibrosis gene for purposes of your work?
No.
Why not?
Although there is a lot of variation in the cystic fibrosis gene, that variation isn't widespread enough in the population so that each time you used it, you would expect to get very much information. It wouldn't be sufficiently informative for forensic science purposes to use it.
So is it correct then that the genetic markers you're most interested in are those that show differences between people and can be easily used and are able to be typed, for instance, in older samples?
That would be right.
Now let's go back to PCR itself. Does it have uses outside forensic science, or is it just limited to work in an area such as yours, like human identification?
PCR is sort of the next thing that came along after the Southern blot that is used everywhere. The use of PCR in forensic science would be a very tiny fraction of its total utility to biological research.
What are some of the uses of PCR outside forensics?
PCR is used in the same kind, in genetic analysis, in gene mapping, and it's used in many basic research settings simply because it allows you to get a whole lot of DNA from a very small amount of starting material. And whenever you can get, scientifically, whenever you can get a lot of something, it gives you the ability to study it. So if you want to look at a particular section of DNA, then to get a lot of it makes your work a lot easier.
Now, you raised the term, or you used the term, genetic diagnosis. Is that determining whether an individual, for instance, either has a genetic disease or is, I think you used the term carrier?
Yes.
What is a carrier, just briefly?
That is, if a parent has a genetic disorder, then the parent can transmit that genetic disorder to his or her children. And this is something that many parents want to be aware of before they start having children. And, so you might say, a family might decide, will we have children or will we adopt children? Because one or the other of the parents could be the carrier of a genetic problem.
Is PCR used, for example, to counsel parents, as you just described, about the likelihood of a child of theirs having a genetic disease?
Yes.
Is PCR used in the area of plant genetics?
I'm sure it is. I'm not a good enough plant biologist to know any specific applications, but I have no doubt that it's being used.
You used the term gene mapping. Can you just tell us a little bit about that?
In terms of understanding human diseases and human genetics, one of the primary things that scientists need to do or want to understand is how are genes located relative to each other. What gene is on what chromosome, what genes reside close to it. And so in the process of answering these questions for specific genes, PCR would be a very helpful technique.
Is PCR used, for instance, in the identification of the remains of American war dead?
Yes.
All right, ladies and gentlemen, we're going to use this point to take a brief court reporter recess for the morning. Please remember all my admonitions to you. Don't discuss the case amongst yourselves, form any opinions about the case, conduct any deliberations till the matter has been submitted to you, or allow anybody to communicate with you. And we will resume at 10 minutes till 11. All right? Dr. Cotton, you can step down.
Thank you, Your Honor. Dr. Cotton, you've described a little bit about the use of these tests, whether RFLP or PCR, to exclude people or include people. Is that right?
That's right.
Is there any difference between those two things, excluding someone or including someone?
Well, those are two different, entirely different things, but I'm not sure what you mean.
All right. Well, first of all, in excluding a person, what does that mean once you've conducted the test?
You're saying that that person cannot be a contributor to the sample that you've tested.
What about the opposite? What about including someone?
Including someone, you're saying that this person could be a contributor to the sample that you've tested.
Is there, in terms of the use of these tests, are they capable and do they in fact serve both of those purposes?
Yes, they do.
Now, as far as the use of PCR, and you've described the fact that there are genetic markers or various locations on this DNA molecule that you look at where people differ,
That's right.
and you described a little bit about the selection process, the process whereby forensic science decides which markers to look at. Is that right?
That's right.
Are there specific genetic markers that you look at in your laboratory following PCR amplification?
Of course.
Okay, do they have names that describe them?
Yes, they do.
Could you describe those for the jury, please?
To tell you the truth, so I don't make a mistake, if you want me to list those markers, how about if you hand me one of my notebooks so I
Sure.
can read off of that?
Would that assist you in describing
think it
each one
would.
exactly?
Yes.
First of all, I'm sorry. Go ahead. First of all, Dr. Cotton, these what appear to be two binders. Can you just tell us what they are?
The two binders have copies in them of our original case folder. I have the original case folder in my briefcase, but these are easier to manipulate.
In other words, those are easier for you to work with?
That's right.
Actually, let me approach it slightly differently. Is there a marker that you examine using the PCR process called DQ alpha?
Yes.
What is DQ alpha?
DQ alpha is located on chromosome 6, and it is one of the human leukocyte antigen markers. These microphones are doing funny things.
Okay.
Why don't you take off the wireless? Just hand it to me. Thank you.
Now, you used a fairly large term, human leukocyte antigen?
That is the name, the generic name of the set of genes for which DQ alpha is one. They are they code for proteins that are on the surface of cells, and DQ alpha is one of these proteins that's been very well characterized, and the DNA that encodes this protein is also very well characterized.
Dr. Cotton, could you pull that microphone just a little closer to you?
Did you
Okay.
disconnect the course microphone?
Yes, Your Honor, the wireless was replaced in that same system.
Take a recess. Fix it. right, let the record reflect we've been rejoined by all the members of our jury panel. Ladies and gentlemen, my apologies to you for that short delay, but you need to hear what's going on. And let's proceed. Mr. Clarke.
Thank you, Your Honor. Dr. Cotton, I believe you were about to discuss this marker called DQ alpha.
That's right.
And is that one of the markers that you type samples at following the use of this PCR copying process?
Yes, we did.
What can you tell us about DQ alpha? How well does it tell people apart?
DQ alpha has 6 alleles. That is, there's 6 different forms of this DQ alpha gene. And with that many alleles or forms, you can generate 21 different types in people. That is, so you're not confused, we each have 2 forms of any genetic locus or any gene. One comes from mother, one comes from father. So you're looking at how many combinations of these 6 things would exist, and there are 21 different combinations of the 6 alleles for this gene, DQ alpha.
Is there a particular term used in science to describe what a person's type is when they have 2 forms of a particular genetic marker?
Yes.
What's that called?
I mean, what I think you're talking about are the terms homozygous and heterozygous.
All right, well, let's start with that first. What do those terms mean? Are they important? Do we need to know those terms?
Do you need to know? Well, I don't know if you need to know them. Probably you do. When you have inherited from both parents the same form of a gene, that is, so both your copy that you got from your mother and the copy you got from your father are identical, then you would be said to be homozygous, homo meaning the same, and zygous referring to a zygote, which is a combination of egg and sperm. So if you have 2 forms, you will always have 2 forms of the gene, and if they happen to be the same, let's use a very easy example. If you got a gene for blood group A from your mother and you got a gene for blood group A from your father, you would have 2 genes, both are for blood group A, and you would be homozygous for that A.
All right, is there also a term called genotype?
Genotype simply, it just means what types do you have? Do you have an A and an A or an A and a B? That's your genotype, AA or AB.
As far as this marker DQ alpha is concerned, then, if everyone in the courtroom were tested, would each of us be one of these 21 different types or genotypes?
We would each be one of those 21 types.
Does that mean, because there are presumably more than 21 people in this courtroom, does that mean that it's likely or necessarily true that at least 2 of us have the same genotype?
Well, probably for those genotypes or combinations of genes that are common, we'd find more than one person in the courtroom who had the same type.
That was actually going to be my next question. As far as these types that we each have, are they distributed absolutely equally or not?
They're not.
Why is that?
I don't think we can. First of all, I'm not sure I can answer that question. That may be an evolutionary answer, and I'm not that knowledgeable about the DQ alpha gene. However, what I know is that the types are not distributed evenly. So some types are very rare, some types are relatively rare, and some types are very common.
We'll return to it later, but are there studies done to determine how common or how rare these individual types are?
There actually is a very large amount of work published on how common or rare particular DQ alpha types are in many different racial or ethnic groups.
Still with this DQ alpha marker, how do you know it's a good marker to use in forensics?
Well, if you use as your judge the first criteria that you want something to be relatively variable, that is, A marker that shows a lot of variation is more useful than a marker that doesn't show very much variation. So on that criteria, DQ alpha is quite good because with 6 alleles giving you 21 different types, that's a fair amount of variation and it's going to have useful information. The test that has been designed for forensic use for DQ alpha, which uses PCR, only amplifies a very short section of DNA. That means the test is going to work well on samples that are degraded. And so for that reason, it's also very good.
Is it, for instance, more useful than the ABO blood grouping types that this jury has already heard about?
Yes, it would be.
Why is it more useful?
It has more variation.
In what way?
Well, with ABO blood groups, you have 3 choices of alleles. You can be you can be O, or you can be B. And that gives you a combination of 6 different genotypes, that is, taking those 3 things 2 at a time. So that means each one of us will be one of those 6 types, as opposed to DQ alpha, where each one of us would be one of these 21 types. So DQ alpha is going to be able to distinguish us one from the other better than ABO.
Incidentally, of these 6 types for ABO you've described, are they actually fewer than that can be actually determined from testing?
With normal serology testing, you can't distinguish all those types. People are developing DNA tests in which you will be able to distinguish all of those types.
All right. In addition to DQ alpha, do you type other genetic markers following the use of PCR?
Yes.
And are they collected? First of all, how many other markers do you type in the laboratory?
In our laboratory, we're typing 5 other markers. Actually, recently we've added 3 more, but they were not done here and we don't need to worry about those. So for purposes of this case, we typed 5 additional markers besides the DQ alpha.
Are those 5 markers collectively known by any term?
The 5 markers are collectively referred to as polymarker, or PM, and that's simply the name given to that's not a scientific name really, it's just the company who developed this test refers to it as PM, generally standing for polymarker.
And are there different ways of describing those 5 different markers that are part of the polymarker system? In other words, are they labeled by a particular name?
Yes, of the 5 genetic locations that are tested in this polymarker system, each one has a specific name.
And can you briefly describe what each of those 5 are in terms of their name?
Sure, but this is what I'm going to take out of my notebook here.
All right.
So.
All right, Mr. Neufeld, do you want to see that?
I'm just going to read this section here. That I can do too.
All right, Mr. Clarke, proceed.
Yes, thank you. Dr. Cotton, what are those five genetic markers?
Each one of them has a short abbreviation which stands for the longer name. If you just focus on the abbreviation, that's probably a lot easier. The 5 markers are LDLR, standing for low-density lipoprotein receptor.
Can we stick with LDLR?
You want me to just give the initials?
That's fine.
Okay. The other is GYPA. The third is HBGG. The fourth is D7S8. And the 5th is GC.
Now, of those 5 you just described, one of them had that notation of the letter D with a number. Is that right?
That's right.
Would that then be another one of those markers where its function in the body is unknown?
As far as I know, it is, yes.
And then the other 4 were described by letters, basically. Is that right?
The other 4 are actually genes, and they have, the letters are just an abbreviation for the name of the protein that's been very well described for that gene.
Now, this set of 5 markers that are part of polymarker, how do you know they're good to be used or appropriate to be used in forensic work?
Each one of these markers does not have a lot of variation, but grouped together, it still is informative. And really the answer to your question is, in addition to the theoretical idea of how informative is this marker, this test has been designed to be also useful specifically for forensic purposes. The sections of DNA that are amplified are small, and the typing is on the dot blot strips that you saw on the tray a while ago. So it's a test that's easy to do in the laboratory, and it works on and has been shown to work on a very wide variety of samples. So it's applicable to all kinds of samples that come in terms of forensic casework.
Incidentally, that tray with the strips that you identified and was shown to the jury, was that actually a polymarker set of typing strips?
Yes, the strips that were in the tray when you looked at them have the 5 polymarker loci, and if you were looking at the strip, you would see that there were letters on the top of each one, and it's the LDLR, GYPA, indicating the dots in that set of this, in that portion of the strip are for that particular genetic location.
All right, referring to Peoples 253.
Yes, thank you, Your Honor. Going back to DQ alpha for a moment, how long has it been used in forensic casework?
I think since about 1986.
Going forward then to the polymarker genetic markers, when were they begun to be used in forensic casework?
We started using the polymarker test in January of 1994. There probably are some other labs who were using it earlier than we do, but I don't know exactly how. It wouldn't have been a lot earlier, somewhere maybe within a year or so before we did. So let's say it's been used in forensic casework since approximately 1993.
Is there some process that these genetic markers undergo before they're actually used in casework, or does someone just discover a gene and start using it immediately?
There's actually a what usually ends up being a very long process of development of the marker, and then what's referred to as validation. That is, if another laboratory uses a marker, that doesn't necessarily mean that I can just bring it into my lab and start using it. So each lab that uses a particular test generally goes through a set of experiments in their laboratory to make sure that in the hands of the people in that laboratory, the test functions as it's supposed to. It meets its specifications, and that everybody that's using it understands what it is they're doing. So there's development of the marker, then there's validation of the marker, which may occur in many different labs, and then there's training the staff in a particular lab to use that marker.
And do you ensure that that marker can be correctly typed in evidence-type samples before you even begin casework use of them?
Of course.
As far as these well, let's start with DQ alpha. Is DQ alpha a marker that's tested, and I think you said more than one laboratory uses the polymarker system. What about the DQ alpha system?
The same is true of the DQ alpha system. It's used by many forensic laboratories around the country and really around the world.
That was going to be my next question. To your knowledge, are the polymarker and DQ alpha genetic markers used worldwide?
They are.
You touched a little bit about, as far as the 5 polymarker genetic markers, that their variation is not as great as DQ alpha.
That's right.
Without getting into each one individually, can you tell us briefly how much variation they each show?
The 5 locations that make up the polymarker system, all of those 5 locations either have 2 or 3 alleles or forms, and they all are designated A or B, and if they have 3 forms in the kit, they're designated A, B, or C. So you can, for example, if you have a marker that has an A type and a B type, any given individual then could have 2 A's, they would be an AA. They could have 2 B's, they would be a BB, or they could have an A and a B, and they would be an AB. So if you have two alleles, the most number of genotypes you can have is 3.
DQ alpha and these polymarker genetic markers, do they represent sequence differences or length differences as you described them earlier today?
These are all sequence differences.
As far as the actual typing of these DQ alpha and polymarker genetic markers, do you actually conduct these tests from scratch in terms of materials and what you use to be able to type samples?
No.
How do you do it?
These tests, the DQ alpha and the polymarker, come as a kit, which means you're buying in a box all the things that you need, or almost all the things you need to do the test. You'd end up making a few of solutions in the laboratory, but the strips come in the kit. The chemicals that you need to do the reaction to create the blue dots come in the kit. The tray comes, you have to buy the tray and the kit. I guess actually you buy them separately. And the PCR mix that contains the primers and the polymerase and the A's, G's, T's, and C's. All that comes in the kit. So you're just supplying some very simple to make solutions and using the kit to do the test.
When you conduct the more powerful RFLP typing technique, do you use kits to do that?
No.
Why is there a difference between the two approaches to DNA typing as far as kits are concerned?
Well, somebody took the time to make the kit for DQ alpha and polymarker, and nobody has tried to market a kit for RFLP testing. I guess you could, but it would have an awful lot of components.
Is there anything unusual about using kits to conduct testing?
Oh, no,
Why do you use a kit?
because it's there.
Does it have
It's a
any-
good test, and it's available, and it's had an enormous amount of work done on it. If it wasn't a good test, you wouldn't use it. If it wasn't available, obviously, you couldn't use it. So the company that makes the kit has put a lot of work into it, and it happens to be very, very good.
All right, turning your attention, if I can, to the last genetic marker that we'll talk about, D1S80. Does that represent a genetic marker that's typed based on length differences as opposed to sequence differences?
Yes.
Was D1S80 developed as a marker to be used in forensics, or did it have some different origin?
As far as I know, it wasn't discovered. I mean, it was discovered, but then basically it was developed specifically for forensic use. I really don't know if there's any research use for D1S80 or not.
Outside forensics?
Right.
How are you familiar with this particular genetic marker?
We've run this marker in our lab. We're not currently using it in our routine casework, but we've run it in our lab. And I've also actually read a number of papers that had to do with D1S80 and heard a lot about it at various scientific meetings.
Why don't you use it in your casework in the laboratory?
The reason we're not using it in our casework is not because it's not a good marker. It's a very practical reason, and that is the kind of gel that the D1S80 is run on is not a kind of gel that we would run other markers on. And so essentially we aren't using it because it's a fair amount of work to do it, and you get one piece of information off, that is the D1S80 piece of information. There are some other markers out there that we chose to use instead. They're not quite as informative as D1S80, but I can do more of them at a time. And so it was a very practical decision that you would just make in the lab to say, do we want to do test A or test B? And has nothing to do with whether or not D1S80 is a good marker. It was just our choice to go with another type of marker.
So there are practical considerations that go into each laboratory's decision of what markers they're going to test?
That's right. And because not every laboratory that does casework has the same type of casework or the same casework demands. And so all of those things go into your decision about what markers you're going to be using.
As far as your familiarity with this D1S80 marker, what type of variation does it show?
The D1S80 has over at least 24 alleles, that is, at least 24 forms. I haven't calculated how many genotypes that could give you, but you can imagine at least 24 things taken two at a time. You can have many, many, many combinations. So it's a very informative marker, and it's, except for the gel system, which takes some pains to do, it's a very good one.
All right, I'd like to shift topics if we can and ask you, are you familiar with the term contamination?
Certainly.
What does that term mean to you?
It means so many different things that maybe I should try to break them down in
All right.
terms of what it means to me. If you think of a biological sample, say you take a blood sample from someone in sterile conditions, you have a very clean, sterile sample. That's sort of the ultimate in terms of having a good sample. As soon as you take that sample out of a sterile condition and say you have a blood stain on a piece of cloth, then you could think about, well, that blood stain is no longer sterile, and it could be, quote, contaminated by anything that's on that cloth. So that would sort of be a second, or let's say a first level. Now, in terms of analyzing evidence from a crime scene, you're always getting things that are like that. You're never being presented with a sterile sample. So in terms of a crime scene, contamination would mean that the sample is that is, in retrieving the sample, it is as it was deposited, that nothing's been added to that sample in the process of picking it up, taking it to the crime lab, and so forth. And then you can also say, well, I have my sample from the crime scene. It came to the crime lab in exactly the same condition that it was at the crime scene, and then clearly you need to be concerned with whether or not in the process of working with that sample in the laboratory, are you introducing any contamination from any source in the laboratory? So you can't use it very well as a generic term. I mean, you could, but unless you specify what kind of contamination you're talking about, then it becomes very difficult to discuss, because you can break it down and you can have many different kinds of contamination, some of which are inherent in the fact that you have a piece of evidence and not a sterile blood sample, and some of which are not inherent and that could occur along the way in how that sample is handled.
As a forensic scientist, are you concerned about contamination?
Of course.
Why?
The idea of analyzing a piece of evidence is that you learn something about that piece of evidence. You're not interested in learning about, that is, if you contaminate that piece of evidence with, let's talk about, say you contaminate a piece of evidence with some other biological sample or some DNA in the laboratory, that will then interfere with your being able to come to a valid conclusion about that piece of evidence. And so you would certainly prefer not to have anything interfere with coming to a valid conclusion about a specific piece of evidence.
As far as this area of contamination, and you've described the two basic DNA typing approaches that you use, both RFLP and PCR, is your concern about contamination the same for both of those techniques, or is it different?
It's different.
Why?
The PCR test is much more sensitive. You can do a PCR test with a very minute amount of starting material. So you would be more concerned. It doesn't mean you're not concerned with contamination for RFLP, but you're going to be more concerned with contamination when you're dealing with doing a PCR test on a piece of evidence.
Okay, let's take RFLP to start with. What steps, if any, do you take to deal with the problem or the potential problem of contamination in that testing process?
Okay, and my answer is going to then be what we're doing in the laboratory, because we're simply receiving a piece of evidence from outside. That is, we're not collecting the evidence ourselves. In the laboratory, our work is done in it's called a biological safety cabinet, but basically it's a setup where you have a piece of glass and you have a working area, and the air is circulated within that working area to both protect what you're working on and to protect yourself. So you have some containment in the area that you're working with these samples. In our laboratory, when a sample is moved from one tube to another, that is, the tube has a label, it has a case number label and a sample number label. And in the process of doing DNA extractions and doing this testing, there are points where you have to remove the sample from one tube and put it into another. That transfer, the labeling on the tubes is witnessed. That is, somebody's checking to make sure that if I'm transferring from a tube that's marked 02, that I'm transferring it. The second tube I have in my hand is also marked 02. The other main precaution is that we do the DNA extractions for evidence samples as opposed to known samples. That is, when I say known, I mean you have a standard sample from a known individual. Those extractions are not done they're done in this hood, but they're not done at the same time. So the knowns and the evidence samples are not worked on for purposes of DNA extraction at the same time.
So those are the primary steps undertaken with regard to the potential for contamination as far as RFLP typing is concerned?
That's right.
What about PCR? Do you undertake any different steps or additional steps to deal with this potential problem?
There are many additional features of how you do a PCR test to deal with minimizing any problems with contamination.
Could you describe those, and are there general categories that you can place them into as far as the steps that you take?
I'm not sure what you're asking.
Well, what are the steps you take to deal with this potential problem?
Okay. Let me think about this a second so it comes out organized. The DNA extractions for PCR are done in a separate location than the extractions for RFLP. Now, in our lab, it happens to be that there's a separate one of these biological safety cabinets that's only used for PCR extractions. And the reason for that is that if you're doing something, if you have enough DNA to work with RFLP, that's a fair amount of DNA compared to what you might have in a PCR sample. So we don't want to be handling the large quantities of DNA that you would have for RFLP in the same place with the same equipment that you're handling the very small amounts. So most laboratories will have a separate location to do DNA extractions when those DNA extractions are for PCR. And in our lab, it's a separate one of these hoods. The sample is, and in addition to that, for example, in the hood, the pipetman, or the piece of equipment you're using to add things to the sample and take things out of the sample, are specified. That is, you have a pipetman that's used only for DNA, and you have a series of pipetmans that are used for everything else, so that you're not interchanging the piece of equipment that you're using to handle the DNA. You are that concerned. And the tips that you're using for the pipetman are these art tips that have a filter in them, so that you can't get any part of your sample up into the pipetman and then therefore accidentally transfer it to the next thing that you work with.
Now, I believe, did you talk about extraction? I'm sorry, extracting evidence?
Well, basically that's everything I just said relates to the area where the DNA is extracted.
All right. Extraction was the first basic phase of dealing with evidence for purposes of PCR typing, is that correct?
That's right.
Earlier, you described the second major phase involves what's called amplification, this copying process.
That's right.
Do you take any precautions or additional steps to, again, deal with the potential for contamination during this particular phase?
Yes.
What are those?
Well, first, now that you've finished the extraction, you're going to set up the amplification. That is, you go through that process where you have a tube, and in that tube you put your DNA, and you put all the things you need to do the PCR, the primers and the polymerase and so on. That setup is done in a second area, a very clean area, and it's in our lab. It happens to be a very small room, but it could be some other just separate location. So the setup of the PCR reaction is done away from where the DNA extraction is done. Once that reaction is set up, it's then carried to yet a third location where the thermal cycler is, where the amplification takes place. Once it's gone into that area where the amplification takes place, it does not ever go back in the other direction.
Why is that?
Think about, in terms of the PCR, you have started with a tiny amount of material, and now, after the process of amplification, you've generated millions of copies of that tiny amount of material. The biggest contamination problem that can occur with PCR is transferring some of that amplified product back to where you're starting out with, because that amplified product will amplify really well if it gets into anything else. So it's not the only concern, but the biggest contamination concern is that you do not get any of that amplified product, which is now a lot of DNA, back into anything that you're ever starting out with. So the DNA samples go from the extraction area to the setup area to the area where the thermal cycler is, and where you then do the analysis of the amplified product, and they do not go back in the other direction.
And that's to, again, avoid this problem of lots of DNA being mixed in or commingled with small amounts of DNA.
That's right.
Now, as far as, and I believe you described the fact that evidence is not extracted at the same time as known samples, or did you mention that?
I did mention that with regard to RFLP testing, and that same general procedure, that is, you don't do the DNA extractions for evidence and knowns at the same time. That's just a general precaution we use in our laboratory, and it's applied to everything.
So as far as RFLP typing, your primary steps to avoid contamination are extracting samples at a different time, that is knowns versus evidence, is that right?
That's right.
As well as witnessing of labeling samples, is that correct?
That's right.
And are those the two primary steps taken as far as RFLP testing is concerned?
That's right.
When you turn to PCR, you utilize those same two precautions, is that right?
You utilize those same two precautions and the other long list of precautions that I just talked about.
As far as this case, and you described a little bit about the amplification phase, was it correct that evidence samples in this case were amplified, or did it happen that they were amplified on a different date from known samples?
In this case, it happened that that's the way the testing was done. Not only were the DNA extractions done at a different time, but the amplifications, that is putting the tubes in the thermal cycler and allowing the amplification to take place, that also was done at a different time for the knowns and the evidence samples. That's not necessarily the way every case is worked. It just happened to be the way this case was worked, really based on when we received samples and when we did each one.
Does that play any role in the potential for contamination at all?
Well, it certainly acts as a preventative measure.
But it's not something that you deem to be routinely necessary.
That's right.
As far as these precautions, and let's focus on PCR for the moment, the precautions that you've described that you use in the laboratory, are they unique to forensic science, or are they used in other areas of science involving PCR?
They are definitely not unique to forensic science, because every lab that's using PCR, no matter what the reason, is still doing this same thing. They're taking a small amount of DNA and making millions of copies of it. So any laboratory that uses PCR for any reason has got to be concerned with making sure this amplified DNA that they get at the end doesn't get transferred back to any place along from the start of the sample on forward.
Are these measures taken, for instance, in laboratories diagnosing diseases,
Certainly.
or in research laboratories,
In research laboratories also.
or in laboratories conducting paternity case evaluations or testing?
Exactly. And I don't mean to imply that, for example, a research laboratory would take exactly the same precautions that we would. They may be slightly different, but no matter what the setting is for PCR, you have to be concerned about making sure amplified product doesn't get back to your starting point.
I'm sorry, could I have just a moment, Your Honor? So in terms of these, and just in terms of summarizing these precautions that you take for PCR, they include the way you handle evidence, is that right?
Yes?
The way you extract evidence in terms of when it's done at a same or a different time than known samples in a case.
Yes?
It includes precautions taken with regard to instruments that you use,
Yes?
as well as the direction of flow, so to speak, of a sample going in one direction only, as you described, and not backwards.
That's right.
As well as, and incidentally, are there other steps taken that, to your knowledge, we will discuss later as well, in terms of dealing with the potential problems of contamination?
Yes.
Now, I'd like to shift topics a little bit to specific forensic case samples that are encountered. And in particular, with regard to evidence, can evidence at, for instance, crime scenes be subjected to basically the environment and the elements in that environment?
I don't think it's a question of can. I think it's a question of that's absolutely bound to happen.
Okay, what types of things can happen, for instance, to a piece of evidence found outdoors?
Well, it's going to be exposed to whatever temperatures are outdoors, whatever lighting conditions. There may be a lot of sunlight on that day. Could get rained or snowed on. I mean, if it's outside, it's going to be exposed to whatever's going on outside from the time that it was left there until the time that it's picked up.
Incidentally, would the same be true, for instance, of a soldier killed in battle out in a battlefield?
I would presume so.
Or dead animals?
Yeah, dead animals too.
You've described a little bit about, for instance, heat. Would that represent one instance of an environmental effect or something that's possible that happens outside?
Sure.
And would that include sunlight as well?
Yes.
And I think you mentioned rain? Or
Well, I
perhaps
mean,
thought-
you said outside, so
Okay.
well, it could get rain. I mean, in California, maybe not, but
Sometimes, some seasons it does.
You'd be surprised.
How about humidity? Is that something else that happens?
of course.
Are there other types of influences that are out there outside?
Well, I think you've named the major ones.
With respect to these influences or effects like heat, humidity, rain, and so forth, what do they do to DNA? What's their effect on DNA?
Basically, all of those things over time will act to gradually degrade the DNA, that is, break it up. The generalization that most everybody is aware of is that dry and cold works to